the primary stain, all cells would take this up. So if you have a mixture of gram-positive and gram-negative cells, they all stain purple initially. Okay. You then wash off excess. You add the mordant, 60 seconds again. Okay. 60 seconds, 60 seconds. That mordant helps the primary stain, the crystal violet, to attach more firmly. Since the crystal violet really can adhere to the peptidoglycan, the mordant is really effective on the gram-positive cells, which have this thick peptidoglycan layer, not so effective with the gram-negative cells. Okay. Now we rinse off excess. You're always rinsing in between these steps. Okay. Rinse. Rinse. Then you add the decolorizer, and for this, there's no set time amount. You are adding the decolorizer while you're holding your slide at a 45-degree angle. And you do it until the runoff appears clear. And usually it's just a few seconds. That decolorizer should be ideally working by removing the crystal violet from the gram-negative cells, because that mordant didn't do its job here. But since the mordant helped the crystal violet attach more strongly here, the decolorizer doesn't do much. So it rinses off the crystal violet from here, but not so much from here. Okay? You do that for a few seconds until the runoff is clear, and then you immediately rinse. As soon as you see the runoff clear, you rinse. Okay, so don't put it down and say, oh, what do I do next? And kind of stop and think. You immediately rinse, because that stops the process. That stops the decolorization. Finally, you add the counter stain. Okay, this again, 60 seconds. Now I mentioned when you add that decolorizer, it would decolorize the gram-negative cells, but not the gram-positive cells. So at this point, after the decolorizer, the gram-negative cells would be clear, whereas these would still have that crystal violet, so they'd be purple. So now when you add that counter stain, these ones can still take that counter stain up, because red is lighter than purple, so it wouldn't do anything on this. That purple is going to outcompete the red. It's darker. It doesn't want that red stain. Okay, so this remains purple, whereas this one was just clear. It was decolorized, and so it can take up that red stain. Okay. Once you finish staining, you're then going to look at it under the microscope and look at the color. If it's red, you know it's gram-negative, but you also know a few characteristics about its cell wall. You know that it has a thin peptidoglycan layer. You know it has this outer membrane. You also know it has LPS and porins. Okay. If it stains purple, you know it's gram-positive. Again, you know some general characteristics about the cell wall, that it has a thick peptidoglycan layer. It does not have an outer membrane. It has these teichoic acids that hold the peptidoglycan to the plasma membrane. And then you can also say that it doesn't have LPS or porins. You could say penicillin is more effective on gram-positive because it targets the peptidoglycan layer. So once you look at your slides, after you finish your gram stain, you are going to record gram-positive or gram-negative, right? Is it purple? Is it red? What is your conclusion based on that? But then we should also be able to reconfirm that shape and arrangement, just as you did on Tuesday. You have your results on Tuesday, from Tuesday, make sure they line up here. And again, at this point in the semester, we are no longer saying circular, rod-like clusters. Instead, we're only using terms of, cocci, bacilli, vibrio, coccobacilli, and then for arrangement, diplo, mono, staphylo, strepto. Okay. Any questions on that? Yeah. Go. So Tuesday was a simple stain. Here is a differential stain. So you can you should have the same results for shape and arrangement, but then you're adding to that with the Graham reaction. Okay? Yeah, Krista?
Between the step of decolorizing and adding the solid, if we see that the gram-positive light is taken on the purple or green, do you also dye that with the counter stain, or do you just dye the one that's decolorized?
You always complete all steps.
You always do.
Sometimes your smear is just the perfect amount that you can tell from your slide, oh yeah, this is gonna be a gram-positive result. But you always want to double check by going all the way through. Sometimes you can't see what's there, kind of like with Asarius.
Yeah.
And then sometimes too, you'll see that once you look at it under the microscope, you may have a mixture. And we'll talk about that, like why is the majority purple, but then we have this little bit of pink here.
Alright, so you always
Go all the way through.
even if you have a clear purple result. Okay, thank you.
So unlike Tuesday's lab, we're staining all three with purple first?
For the most part.
Yes, all three are getting stained the same exact way with the gram stain. Yep, so purple first. Any other questions? Okay, the last thing that to do is that on the side bench there, you have some demo scopes. We are not going to carry through these special stains, like an acid-fast stain and a spore capsule stain, but we do want you to just take a look and see what they look like. So acid-fast, these are a special type of bacteria that have this extra layer outside of it with mycolic acid. They're especially resistant to staining as well as disinfectants and stuff. One example of an acid-fast bacteria is the one that causes tuberculosis, okay? And so in order to stain these, to provide some kind of contrast so you can look at them under the microscope, there's a special technique. And there was a virtual lab that was optional in case you wanted to explore it online. These ones, basically, you need to have some special method of getting a stain into the acid-fast bacteria. So it could be heat that helps drive it in, but then once it's in there, it's kind of locked in. So when you add a decolorizer, it doesn't remove it. But the decolorizer does work on the non-acid-fast bacteria. So you have a differential stain where you're adding one stain with heat that drives into all cells, then you decolorize it, but the decolorizer only works on the non-acid-fast because the acid-fast ones retain it. Then you add a counter stain that the non-acid-fast take up. Okay? So when you look at it, you'll see that it almost looks like a mixture of gram-positive and gram-negative because you have pink and you have blue instead of purple. But you would have to know what was the technique that was carried out. Oh, it's acid-fast stain here. Therefore, anything that's pink is an acid-fast bacteria. Then to look at an endospore stain, we've talked about endospores in last lecture. Note that the endospores themselves can form at different locations in the cell, right? When they are forming through the process of sporulation before the mother cell degrades, they can, certain species create them at the end of the cell, these are terminal endospores. Some create them in the center of the cell. Again, this is before the mother cell breaks apart. And so take a look at that. If we were to try to stain these with a gram stain, the spores would not take them up. So they would just look unstained, kind of like that homework assignment I had for you on anthrax, where it's just unstained. You try to stain it with either a simple stain or a gram stain, and it doesn't take it up because the endospores have all those protective layers. But if we carry out a special endospore stain, kind of like with the acid-fast, again, you can use heat to drive it into the spore and stain the spore. And so it's a pretty faint color, unfortunately, over there, but you can see that the spores are green, and then the vegetative cells are red, saffron. And then the last one is a capsule. So some species have capsules. These are on the outside of the cell wall, and they protect the bacteria from phagocytosis. So it's really hard for certain cells, like our white blood cells, to gobble them up, to phagocytose them, if they have this capsule. The capsule is really challenging to stain, not because it's so resistant to anything coming in or out, but because it's so water soluble. So when you rinse it, it rinses the stain out of the capsule, okay? The stain remains inside that inner part of the cell though. So So the plasma membrane, all its contents will stain, and it'll retain its stain even when you rinse it, but the rinse washes it out of the capsule. So it's kind of neat to look at because it looks like